Monday, there will be a quiz over the Anki cards that I put up yesterday. So I may put up more this weekend, but it's only the stuff from yesterday, which is chapter 5, I think, material. Has a lot to do with the polio vaccine development and that sort of thing. For next week, the immune book that we talked about in the hour earlier than this, read chapters 17 through 23. So it's the last part of that section that it's covering. And then finish chapter 7 in Allen for Monday. Realistically, we probably won't finish that till Wednesday, but going to keep ahead stuff. All right, so the content for this week was related to the lymphatic system. Primarily, we're going to be focusing on the secondary lymphatic tissue. What were the organs that were considered primary lymphatic organs? Thymus was one. And the bone marrow. What's special about the thymus and the bone marrow? The bone marrow is where what's produced? B and T cells. And the thymus is where T cells mature. So this is really has to do with the initial development of the systems. The secondary lymphoid tissues are going to include which ones? Spleen. The lymph nodes, which would include your tonsils. And the last one are the mucosal associated lymphoid tissues, or the MALTs. So these are the lymphoid tissues that are associated with your mucosal surfaces, such as the Peyer's patches. Within the lymph nodes, we're going to talk about the sort of structures that are found in them. So first of all, within lymph nodes and other secondary lymphoid tissues, organs, you have the lymphoid follicles. So the lymphoid follicles are really not well separated from the rest of the lymph tissues, but they are kind of denoted by the presence of what? No, not P, F. That'll make more sense. F stands for follicular dendritic cells. So these stars represent the follicular dendritic cells. Note that these are different from the other dendritic cells that we have talked about. So they are not the antigen presenting cells that bring the antigens back and actually start activating. Rather, they're going to pick up and retain the complement opsonized antigens. They will present these to the B cells and will help lead to B cell activation. So if we do this, these are the B cells. So the B cells are diffused throughout the area in the lymphoid follicles as well as the surrounding area, but the lymphoid follicles are going to be enriched in these follicular dendritic cells and ultimately are going to be what activate these B cells. So the presentation to the B cells is different from the other dendritic cells. The other dendritic cells are going to present via MHC molecules instead of just opsonized antigens. The B cells that are exposed to the complement system are going to proliferate. So the B cells that are exposed by the dendritic cells, if they recognize the opsonized antigen, will proliferate. As this happens, the follicle grows and becomes where B cells develop. So if you've ever had an infection and noticed that your lymph nodes are swollen, part of this is water retention, but part of it has to do with the proliferation of B cells within the follicles that lead to their growth. So B cells, when they are activated by these follicular dendritic cells, these are going to become the follicles are going to become secondary lymphoid follicles or germinal centers. So what's different about the germinal centers compared to the primary lymphoid tissues is how you have distribution of the cells within there. So we still have our follicular dendritic cells activating. We still have B cells diffuse around, but then we also have this region down here that contain a lot of B cells. So this is divided up into two different zones. You have the light zones. Is that blue showing up or is that hard to read? Okay. And the dark zones. The light zones are going to be places where you have the activation of B cells. And then some other things are going to happen as well. So activated B cells are going to undergo hypersomatic mutation. It's not necessarily that hypersomatic mutation is occurring here in the light zone. It's what happens during this process. So what does hypersomatic mutation lead to? Okay, so the hypersomatic mutation is going to need to have the antibodies tested for affinity. So once they've been tested for affinity, they're going to be selected for if they have higher affinity and not selected for if they have lower affinity. If they have lower affinity, what happens to the B cells? Is that why you're via apoptosis? So they die via apoptosis. If they have higher affinity, then they're selected for and they proliferate more. The dark zone is the site of proliferation. The hypersomatic mutations may actually be happening down here in the dark zones where proliferation is occurring, but then it has to move up into the light zones to be tested for that. As they proliferate, they are going to also have one of two fates. So some leave and will essentially go to fight. They'll go to new locations in your body to secrete antibodies. So what kind of antibodies are they secreting? These are the antibodies that are initially proposed or initially made to fight infections. You were way off. So IgA is the mucosal antibodies that helps with preventing things from moving through the mucus. IgG is more of the blood ones that just go through your blood that have gone through this after antibody switching, class switching has occurred. They generally are better antibodies than the IgM. And then there's the IgE, which are parasitic worms. Your task is to come up with a mnemonic for remembering the different types of antibodies, because I can't do it on the fly right here. Game, but remembering which one is which. Yep.
That's where we got stuck.
Be like, guys, I'm not here first. Neither am I, says A. I don't know. So IgM antibodies, are they good antibodies? Are they bad antibodies?
IgM?
Yeah. Are they great antibodies? The first antibodies that you secrete, are they?
No.
They are good, but not great. The hypersomatic mutation is going to be what leads to great antibodies, because they're going to have higher affinity. The IgM antibodies are good enough to kind of get out there. They're not perfect, but they're something that help with the initial fight. In addition to this, the dark zone is going to have some of them some of the B cells stay. Then they undergo the hypersomatic mutation and class switching. Which then need to go back into the light zone to be tested for that. So let's talk a little bit about the anatomy of a lymph node. So lymph node is more or less bean-shaped. It's almost like it has an outer layer here. It's going to have essentially two regions on the inside and then another area here. So this, we're going to call this area A and this area B. A is the marginal sinus. Why would we call this the marginal sinus? What does it mean for something to be on the margins? They're on the edges, right? Sinus is just kind of like an empty area. So there's not a whole lot in the marginal sinus. This is going to be where lymph goes in. They go from the marginal sinus, which I labeled as A, to C. C, so this is the pathway of lymph through the lymph node. From A, I should have, made sense to go in alphabetical order, but I didn't for whatever reason. That is the B cell area, which is also known as the cortex. So I did that on purpose so that I can make C be the cortex. That was totally not an accident. Then goes to D, which is the T cell area, also known as the paracortex, because it's next to the cortex, right? So the T cell area is where the T cells ultimately will congregate, while the B cell area is where the B cells are going to congregate. And then finally, it's going to go to B, which is the medulla or medullary sinus, and that ultimately is where the lymph will go out. Now, in addition to this, we have the structures that we talked about earlier. So these represent the what? Oh no, I'm lymphoid follicles. I knew they were follicles. I was just trying to come up with something after follicles, and it was before. So these are where the B cells are being activated within there. So that would make sense that that is going to be in the B cell area, right? In addition to this, we are going to have blood vessels that are present as well. So the blood vessels are going to come in. Oh, I shouldn't have done that. So this, let's say, is the arterial. Arteries do what? Okay. And this is the venule. Veins do what? Okay. Right. The little dot here that I have drawn on the end of it represents an HEV, or high endothelial venule. The high endothelial venule is essentially a doorway for the lymphocytes. The high endothelial venule is going to be found in all secondary lymph organs except the spleen. The spleen is different because it's going to get its B and T cells directly from the blood because it's targeting more the blood-borne pathogens than pathogens that have been traveling via the lymph. And so the venule then is going to be how the B and T cells get into the lymph. So they're going to start off in the T cell area. Some of them will migrate here to the B cell area, depending on the type of cell they are. So if they're B cells, they're going to go to the B cell area. If they're T cells, they're going to stay in the paracortex. This is accomplished. So the organization is accomplished by cytokines. So there's going to be specific cytokines that are going to interact with receptors. The receptors on the different type of lymphocytes are going to be different, so that the T cells will be attracted to the cytokines in the T cell area, while the B cells will be attracted to the cytokines in the B cell area. Now, even though they are going to be heading towards these two regions, there are some interactions between the T cells and the B cells. Ultimately, the T helper cells help activate B cells, and the B cells help activate the T helper cells. So their stimulation is going to lead to mutual maturation, so that you end up with B cells that become more mature, and you're going to end up with T cells becoming more mature. Ultimately, the T helper cells in this case are going to become follicular T helper cells. Follicular T helper cells are going to help rescue the germinal centers. So unless your body is actively battling an invasion, the germinal centers don't always stay in the secondary state. They'll decay back and form the primary lymphatic follicle structures. The reason for this, you don't need them to actively be proliferating if you don't have an infection. But if you have an active infection, the follicular T helper cells need to keep these things active so that they don't stop maturing B cells and sending mature B cells out to the cells. In addition to this, these are going to help with somatic hypermutation and class switching. So the T helper cells are going to be informed what kind of antigen it is and where that location is, and that's going to help with making the antibodies that are more focused on the types of invaders that are present. Ultimately, if we look at how the T and B cells are going to function or migrate between the lymphoid tissues, the T cells tend to recirculate, whereas the B cells don't. Why would that be? Why would the T cells recirculate and go to different lymphoid tissues? Typically, it's going to recirculate to ones that are similar, because there are differences in the secondary lymphoid tissues once they've been activated. But B cells don't. Why are the T cells going to migrate but not the B cells? What are the T cells doing? What's that H stand for? Helper. They're helper T cells, right? So they send a co-stimulatory signal that helps to activate B cells. And so it goes to the different lymphoid organs and helps activate the different B cells that are there that might be a fit for the type of invader that's present. Meanwhile, the B cells, where do they get activated at? What's that? That's where they're born. Lymph node, where in the lymph node? It's the word that I couldn't think of earlier. The lymphoid follicles. So their site of activation is already present. So they don't need to go somewhere else to get activated. So they might as well just stick around there and then go to the sites of infection. So the T cells will spread around. The B cells, they will be secreting antibodies. They might go to the sites of infection as well. But for the most part, they don't need to circulate to the different lymph nodes because they already had everything they needed to be activated where they're already at. Does that make sense? All right. What's a Peyer's patch?
It's a secondary organ in the small intestine.
Small intestine is a mucosal surface, so it's a mucosal associated lymphoid tissue. So exactly, it's a secondary lymphoid organ found in the small intestine. They're going to have similar functions to the lymph nodes, but they differ in one important way. So they don't have incoming lymph. Why not? What are the lymph nodes trying to sample?
Blood. Tissue fluid?
The blood, the tissue fluid, stuff that's sort of inside the body, right? What's the Peyer's patch trying to sample?
Keep things from getting from the small intestine into the body.
Right, certain things.
Right.
So it's trying to sample things that are found in the small intestine and not necessarily in the spots within your body already. So it's not something inside your body that it's trying to sample. Because remember, you're a tube, so the digestive system is essentially an outside of your body, even though it's found inside your body. So they instead have cells that are known as M cells. So if you'd look at a Peyer's patch, they would be surrounded by something known as an M cell that, so this is the intestine, this is the Peyer's patch. The M cell is going to sample stuff from that specifically and bring it to the Peyer's patch. And so is the M cell going to sample everything?
Well, no.
No.
They're like soft spots, kind of, and something that would be easily attacked.
Right. So they're somewhat selective in what is sampled because there's a lot of things that goes through your digestive system that you don't need to build an immune response against, like food. So the M cells are going to select what goes to the Peyer's patches, and then everything, it does have outgoing lymph. Because it is going to bring in fluid, and lymph is essentially extracellular fluid that is not blood moving through your body. So it does have the outgoing system, but it's essentially going to concentrate things that are potential pathogens and not sample everything in the extracellular environment. All right, the last secondary lymphoid tissue that I want to talk about is the spleen. It differs in that it does not have any lymph going through it. Instead, it is going to sample the blood. When it samples the blood, it's going to then bring in T cells and B cells that way, and not necessarily, and not through the high endothelial venues of the lymph nodes, because it doesn't need those in order to sample stuff. So they have no HEVs because it doesn't need that to bring in the B and T cells. Something that's unique about the spleen is that it will allow for the activation of B cells without T cell help. How does it do this, Jacob? Correct answer is you don't know because nobody knows. Come on. Come on, you gotta say when you don't know things. Gotta be humble. Yes. But why is this a good site for this to happen? What do T cells use to activate B cells? What do they recognize, I should say? Proteins. The problem with some blood-borne pathogens are that they are surrounded by capsules. So this is essentially like a carbohydrate coat that hides the bacteria. If you remember back to Bio 111 when we talked about the R and S strains that Frederick Griffith looked at, one was pathogenic and smooth looking because it had a capsule, and the other one was rough and not smooth looking and non-pathogenic because it lacked the capsule. And so the capsule allows it to avoid immune detection because the T cells can't recognize proteins on there. But B cells can recognize this. So B cells will recognize bacteria with capsules because it does not need the T cell activation to co-stimulate it. This is why certain individuals that have no spleen, because there's been ruptured or were removed for whatever reason, are going to be more susceptible to blood-borne pathogens that have capsules because they don't have an immune system that's as good at fighting these bacteria as others. So the logic of the immune system, essentially the organs, the lymph nodes, are generally going to be located close to the sites of infection that they're fighting. They are going to provide environments that are conducive to sampling the environment and mobilization of weapons and mobilization of the appropriate weapons. And it makes it so that it is a high probability for the adaptive immune system to be activated. Now, when it comes to lymphatic cell trafficking, so that's the T cells and the B cells that are going to come into the lymphatic system. It's going to be different between naive and experienced cells. So naive cells are going to go directly to secondary lymphatic vessels. They can travel to all of them. They're not limited in where they go. This is important because the T cells, they want to be able to go to different places to see what's out there. They want to explore the world. They're young. They want to see the world. The experienced T cells have been activated, so they want to continue going to lymphatic vessels to help activating them, but they go to similar lymphatic organs. So you won't necessarily have an experienced T cell that was activated in your tonsils go to your spleen. You might have an experienced T cell that was activated in one tonsil go to the other tonsil because it's likely going to be a similar type of infection that activated it in those two areas. In addition to this, it also will go to the sites of infection because they can activate cells there where the site of the infection is. So I was talking about T cells. For B cells, it is similar, except you're talking about B cells instead of T cells. Right, questions? I have one question for you guys. Why do mothers kiss their babies?
Sample their the microbes are about to ingest.
And why does the mother want to sample those microbes?
To give antibodies to the microbes into their breast milk.
To essentially give their babies passive immunity towards things that they might be exposed to. It has absolutely nothing to do with love. it's just a biochemical response that triggers, and I don't know. Just a bunch of oxy